Bismillah ar-Rahman ar-Rahim. I'm Abrar working as volunteer at Sin Biotechnological Association. Today I will explain a very important topic entitled Importance of 3D bioprinting and drug discovery. Means that how we can use 3D bioprinting technology in drug discovery process. The objective of this session include the overview of drug discovery and its process, though explain different approaches of drug discovery which are still used are still existing. What is 3D bioprinting, or what is the concept of 3D bioprinting, and what is the importance of 3D bioprinting over the other existing methods of drug discovery. So keep in mind this objective. The contents of this session include the very brief about drug discovery, the traditional methods of drug discovery or the existing method of drug discovery, the concept of 3D bioprinting, what are the processes which are used, and what is the process of using 3D bioprinting in drug discovery, and why we use 3D bioprinting. It's mean that what are the advantages of using 3D bioprinting for drug discovery process, and what are the future perspective of this technology. So just starting formally, very briefly about drug discovery. What is drug discovery process? So the process of identifying a chemical substances which have the potential to become a therapeutic agent in future or treat a disease is called drug discovery process. In nutshell, we can say that drug discovery is a process in which we can identify some substances which have some chemical properties and have some biological effect and use for the treatment of some disease. These are called drug discovery. Why we do drug discovery? Very briefly and very broadly, we can say that drug discovery is mostly performed to treat various kind of existing disease and also the new disease which are coming with time. So how to treat such kind of disease and how we can save ourselves from the causative effect or the illness potential of these disease. So this process for drug and this process to treat the diseases, definitely there must be some potential substances, and how to identify, how to commercialize these substances. This whole process from identification to commercialization is called as drug discovery. There are multiple approaches which are still existing for this therapeutic agent identifications and their commercialization. For example, the first and the oldest one approach is called trial and error method, in which the researcher are trying to identify some substances based on their trial and error method. And the very well-known example is penicillin, which was discovered by this method. The second approach is called chemical modification, in which the researcher are just modify the existing chemical substances which have some biological activity by the addition or deletion or by some chemical reaction into another substances which have the desired properties or desired therapeutic abilities. For example, aspirin is one of the well-known example of this process. Another approach is called high throughput screening or high throughput process. In this approach, the scientists are using to screen virtually available databases by different substances or by different methods, and they are trying to identify a substance or chemical entities which have the potential therapeutic abilities against a certain disease. Beside this, one of the recent and smartest approach toward drug discovery is called as rational drug designing. This is considered the cheapest approach because in this approach, the scientists first identify a biological target, then they identify a hit molecule which can have compatibility with the biological target substances. Then they perform a structure optimization, which lead to the compound having some optimal affinity, have some selectivity, have non-toxicity ability, have solubility in water, also have permeability and bioavailability and other desired properties for drugs. So in this approach, the scientists starting from identification of molecule to the availability and the desired properties of substance to be used to be used as a drug. This whole process, this whole approach is called as rational drug designing. But this, beside this vast majority of available or existing drug discovery process, there are multiple limitation include or multiple limitation existing. For example, the existing method required very extensive laboratory experimental methods. They required very high cost, and also the success rate is not so much high. They have limited productivity, also leading to late stage failure. It means that some of the available drug may be showing potential effect at the beginning, but at the end of at the last stage of clinical trials or post clinical trial, they have maybe some adverse reaction or some negative reaction. So they also have this failure is also associated with the current approaches. These are very much time consuming. At least they can take 12 to 15 years of time, and also inefficient. So it's mean that because of the availability of these limitation or association of these limitation with the traditional methods, the scientists are trying to find some new approach which at least identify or at least compromise all these limitation or few of them. So the scientists just find a very new look toward this drug identification, which is called 3D technology. What is 3D technology? Because why the scientists converted to use this 3D technology for drug discovery? Because it's a faster, more cost-effective, and they also have shortening the drug developmental timeline, a timeline which is almost reduced from 10 to 15 years to very less time. The 3D printing part is it offer a transformative approach to drug discovery because they provide a more realistic and efficient platform for testing and developing pharmaceutical substances. Now, a question arises, we can use 3D bioprinting for drug discovery, but what 3D bioprinting is actually? So very simply, it is considered a very important and cutting edge technology currently, which in this technology, they use a 3D bioprinter or 3D printer and create a living tissues or organs. How they are create these substances by using of some by inks and some by material. So in simple, we can say 3D bioprinting is a technology in which 3D printer are used to create a biological system, a biological organ or tissues by the use of some biological inks and some biomaterial. So these are called 3D bioprinting technology. This technology is also considered a game changer in drug discovery because of its ability to replicate human tissues in organ, because it's important that someone can test their desired compound against their tissues or the organ, and this 3D printing can generate this organ, which mimicking the human physiological responses. It also allow a potential drug compound on 3D printed tissues and reduce the reliance on animal testing and also accelerate the drug developmental process. By providing a more accurate representation of human biology, 3D bioprinting enhances the predictability of drug effect and toxicity. So it's one of the important of 3D bioprinting over other method that they can increase the predictability of drugs and also elaborate and also explain the toxicity of this particular compound. This technology also personalized medicine in use in personalized medicine. So definitely today world is going and the researchers converted to identify or develop personalized medicine called PM. So how 3D bioprinting is also incorporated and also used in this personalized medicine development. What is the process? Very briefly, how 3D bioprinting is used in drug discovery. So very briefly, first they can use as a modeling. For example, the development of realistic disease model. The 3D bioprinting and drug discovery, first the researcher identify a model, for example, cancer, tumor, or organ and chip system. For drug testing, they develop such kind of model and then they test their compound. Then in drug screening, for example, researcher can use 3D bioprinted tissues to screen and evaluate drug candidates more accurately. They can also use to generate tissue. So for tissue regeneration by printing aids and tissue regeneration research is essential for therapies like regenerative medicine and organ transplantation. So 3D printing is also used for tissue regeneration, which can ultimately help to identify suitable drug for the treatment of various diseases. And they can also help in some case studies. For example, 3D printing for liver tissue or for drug metabolism is also, and heart tissue for cardiac drug testing is also already available and also in usage. So it simply means that 3D bioprinting is used for drug discovery process, and there are still some cases available which show its potential effect. Now, why researcher convert to use 3D bioprinting for the drug discovery process? So what are the advantages of 3D bioprinting? First, precision. It means 3D bioprinting allow for precise control over the placement of cell and biomaterial. Definitely, we can we can make, or we can regenerate substances which have some biological, mimicking biological system, biological activity, and we can control this. So it gave definitely a precise result. They can also give enhance the efficiency and streamline the drug discovery by enabling a high throughput screening and reduce the time needed for identifying a chemical potential drug candidates. They can also reduce the cost by replacing costly and timely consuming animal testings in vitro by printed model, for example. So definitely by the help of 3D by printing, the researcher can generate human tissue or organ, and then they perform the routine drug testing process. But as there are multiple beneficial aspects associated with 3D by printing and drug discovery, but this also considered a challenges for the researcher. So what are the challenges and hurdles which are facing by the researcher while using 3D by printing technology for drug discovery? For example, by printing complexity, material compatibility, regulatory framework, scalability, and ethical consideration. So what are these? Let's explain one by one. For example, in bioprinting complexity, the how a bioprinting can mimic or can create exactly same tissue organ that of the human, for example. So it's very complex system and a very complex mechanism in which the bioprinting can make and create a substance or a tissue which we make in the especially the human physiology. The material compatibility, for example, that bioinks and the biomaterial which are used for this process have 3D system are mimicking the natural environments or the natural process also. The most important among them is the regulatory framework, because developing standardized regulatory framework for 3D printing, tissue, and drug testing and fuel is essential, but still evolving. It means that there is no proper and strict guideline which must be followed for the 3D printing and drug discovery. Scalability means scaling up by printing process to meet industrial demand. If, for example, this technology is used widely, and how this technology can enable the research and developmental system that the researcher can create more and more substances by this process. And there are some ethical consideration associated whether this technology is used to create a human tissues or organ, not only for drug discovery, but maybe someone can use this technology for other purposes, illegal purposes also. So these are the challenges which are associated with this technology. So now, what are the future perspective of this technology? Personalized medicine development is already described that personalized medicine is currently widely used concept, and the researchers are devoted to identify substances which are used per every individual. So this technology is also used in this concept. They can also foster drug development, which is also the benefits. For example, this technology can decrease the time per and the time for drug discovery. So this can also be more increased in future. They can also reduce the cost, which is millions of dollars. So they can reduce to very low cost because of this technology in near future also. And these can also novel therapies. Just mean that by the use of this technology, the scientists can convert and can produce more innovative ideas, concepts which are used not only for drug discovery, but can also use for different purposes. The very excellent example for this, for example, one substance is called spartame. A drug which is called spartame is developed by this technology and currently available in the market. So not only spartame which is developed by this tree by printing, other substances or drugs are also available, but this technology may be more promising and have more potential to be used in near future. In conclusion, we can say that 3D by printing stand a transformative force in drug discovery process. It offers precision and efficiency, enable the creation of realistic tissue model for accurate drug testing, collaboration between academia and industry, which is also considered a challenge, is that if this process successfully installed and successfully used, so can academia or industries can fulfill the demand of the society or demand of the population. So collaboration must be developed by this technique, this technology, and also the potential of personalized medicine, which is currently very widely used concept. And this technique and this technology is also used for the development of personalized medicine. These are some references which from which this material were collected, and I'm sure that this lecture will help you at least how the scientists or the researcher use their mind, use their concept, and use the cutting edge technology for various processes. One of them is the use of 3D bioprinting technology and drug discovery, which not only reduce the time but can enhance the efficiency, capability, and reduce the cost also. With this, I'm concluding my lecture. Thank you very much.